networks. Please, Kerr.
Thank you so much for the introduction. So I guess I'll continue again from the start. So hi everyone, nice to meet you all. My name is Kerr and I'm a first-year PhD student from Georgia Tech. It's a great honor to be able to present our work on supervised biological network alignment today. And yeah, I apologize for not being able to present it in person. My co-authors are Dr. Sheng Wang from University of Washington, Seattle, and Dr. Yuna Luo from Georgia Tech. To begin with, I would like to briefly introduce the background of our work and why network alignment matters. So nowadays, with the advances in sequencing technology and protein structure prediction methods, we have accumulated a massive number of protein sequence and structure data. However, well, a large number of the proteins with known sequences and structures remain functionally unannotated. This is largely because the experimental functional annotation of proteins are labor-intensive, time-consuming, and expensive. And especially when it comes to human proteins, it can be extremely challenging or infeasible to carry out experiments on humans due to the technical and ethical concerns. Well, this highlights the importance of translational studies in biomedicine. Researchers have been using model organisms to study the fundamental questions in human disease and clinical applications as a substitute instead of directly performing experiments on humans. Because compared to humans, model organisms are simpler biological systems, they have faster generation cycles, and they can be readily manipulated genetically. And this brought us to a pivotal challenge after we performed the translational studies on organisms. How can we transfer the functional knowledge we learned from the species that we study, for example, the model organisms, to another species that we care about, for example, humans? And this brought us to the major focus of our work, which is biological network alignment. Because protein perform functions by interacting with other proteins, which forms biological pathways and protein-protein interaction networks, proteins that have similar roles in the PPI network often share similar functions. And network alignment refers to the methods which finds the functional node correspondence between two species PPI network. By identifying the topologically similar regions, the network alignment methods can transfer the functional knowledge from one species to another. For example, we can map the yeast protein functions to human proteins via the network alignment methods. And speaking of the topologically similar features, graph-like features have been pioneering some graph features to delineate the topological similarity, and it has been widely applied in the field of network alignment. Here, we have briefly summarized the existing network alignment approaches. Current NA methods can be divided into two major categories: unsupervised NA approaches, which do not use protein function data as supervision, and supervised NA methods, which uses protein function data as supervision to guide the alignment of functionally similar proteins. Well, the most direct unsupervised NA approach would be to perform sequence similarity search, such as using BLAST or MM62. However, previous researchers have found that sequence similar proteins may perform very different functions, and functionally similar proteins does not necessarily have similar sequences. And for unsupervised network alignment methods, which uses PPI networks as their input, their key assumption is that proteins that are in similar topological positions with respect to other proteins in the PPI network tend to share similar functions. Representative algorithms include search algorithms, genetic algorithms, random walk-based, graph-like-based methods, and latent embedding methods. But this key assumption does not always hold due to the PPI's noises and incompleteness. Therefore, to address the previously mentioned challenges, the authors of Tara and Tara++ have proposed the supervised network alignment paradigm, which uses protein function data as supervision signal. During the training, the models are driven to tease topological features that are more informative for network alignment, apart from other distracting signals, say the network noise or incompleteness. The Tara and Tara++ series of work are pioneering in proposing the supervised network alignment paradigm. In our work, we developed Grana, which is a supervised framework for pairwise many-to-many network alignment problems. As the Tara series of work only uses logistic regression, we aim to develop a more advanced deep learning-based solution for network alignment. In short, Grana is an end-to-end graph neural network-based architecture, and the GNN architecture enables Grana to efficiently integrate various sources of heterogeneous data and enables the model to capture high-order nonlinear topological features. Below here is a simplified workflow of Grana, which takes two species PPI network and the non-functional anchor links as input and outputs the functional correspondence between the two species PPI network. Here is a comprehensive overview of the Grana's pipeline. The whole pipeline can be divided into 4 parts, including input data preparation, initial features computation, the graph neural networks module, and the final prediction module. Grana first built an integrated graph as input. The integrated graph incorporates 2 species PPI network and the various types of anchor links. In this work, we mainly explore anchor links of orthologs and sequence-similar pairs. Orthologs are proteins in different species that originated from the same ancestor and are very likely to retain similar functions. And sequence-similar pairs are protein pairs identified by BLAST or MM62. Then, for each PPI network, we build network features of proteins before putting the graph into the graph neural networks. We have applied 2 types of embeddings. We used Laplacian positional encoding for positional embeddings. The key intuition of positional embedding is that the nodes that are close to each other will have similar positional embedding, whereas nodes that are far away from each other will have different embeddings. And we use NetMF for distance embeddings. Unlike the positional embeddings, the NetMF embeddings encodes the similarity between long-distance neighbors. And therefore, the 2 embeddings we have chosen are complementary. Next, the integrated graph with the initialized features would be passed into the graph neural networks. The key of the graph neural network is graph convolution, which is also known as the message passing. So the key idea is that a node first aggregates the features from its neighboring nodes, then updates them with neural network layers, and then send out the updated features to its neighboring nodes. In our work, we have applied a message aggregator adapted from the Jankov. Grana in total has L blocks of graph convolution. So within each block, there are within network propagation layers, which propagates messages solely on PPI edges. And there are across network propagation layers, which propagate the messages along the anchor links. Well, specifically for each type of anchor links, here orthologs and sequence similarity, we each have a unique propagation layer thanks to the use of GNN architecture. Yeah. Finally, for each pair of proteins, we concatenate their representations into a single vector and use the fully connected layer to predict their functional relatedness. The training data and the model's outputs of Grana are sets of functionally related protein pairs. We define the functional relatedness of 2 proteins as whether they share the same gene ontology terms or not. Positive edges refers to protein pairs that share the same geo terms, and negative edges refer to protein pairs that do not share the same geo term. Well, because in Grana, the ground truth data only contains positive edges, we need to sample negative edges for models training. And because nodes that has positive edges are in a very small amount, the traditional random negative sampling might lead to the machine learning model to learn the node's presence in the training data rather than the functional relatedness. Therefore, to encourage the model to learn the functional relatedness, we employ the hard negative sampling strategy that which requires that the sampled negative edges must contain nodes that have both appeared in positive edges. Here is a small illustration. For our data sets, we used 6 species PPI network downloaded from BioGrid. Our orthology data are downloaded from OrthoMCL database, and our sequences are retrieved from the Swiss prod of UniProt KB. As for functional data, we downloaded our functional annotations from the gene ontology, and we only kept annotations related to biological process. Furthermore, to prevent data leakage, we have split our train test data based on nodes instead of on edges. As shown in the right figure, a protein would never appear in both of the training sets and the test sets. We first evaluate GRANA's ability to predict the network alignment between species. The problem can be formulated as a link prediction task, and we have aligned 2 yeast species and aligned the human with 4 other model organisms, which are yeast, mouse, roundworms, and fruit flies. All baseline methods are provided with orthologs as anchor links, and GRANA-O is a variant of GRANA using only orthologs as anchors. We first confirmed the advantages of supervised NA paradigm over unsupervised NA methods. TARA-TS and TARA++ outperforms the best unsupervised methods eTNA in 3 of the 5 NA tasks. Moreover, we have observed that our model, GRANA, consistently outperformed all baseline methods in all 5 NA tasks in terms of AUROC and AUPRC. Then we find out that even with the partial model, the GRANA-O also performed our baseline methods with clear margins. Well, the results here demonstrated that GRANA can better explore the topological similarity to accurately align networks. Next, as the important application of network alignment is to transfer functional knowledge between species, we evaluated whether the network alignment predicted by GRANA can facilitate protein function prediction. For each method, we transferred the functional annotations through top-ranked predicted protein pairs and evaluated the Jaccard index of their functional annotations. So the intuition is that the higher the Jaccard index, the more functional similar are the 2 predicted protein pairs. We observed that GRANA-O outperformed all other methods in 3 of the 4 NA tasks, and our full model, GRANA, further boosted the performances. This experiment showed that GRANA can translate its effective predicted network alignments into the accurate predictions of protein functions. We then performed several ablation studies to analyze our key model designs in GRANA. We included our top unsupervised baseline methods, eTNA, and our top supervised baseline method, TARA-TS, for comparison. Well, as shown in the figure A, we found that GRANA is the only method that can be boosted by the use of heterogeneous various types of anchor links. Well, this is not surprising because of the GNN structures of GRANA, GRANA can represent different anchor links as different types of edges, where eTNA and the TARA-TS are either unable to distinguish between 2 different types of edges or are computationally too expensive for doing that. As shown in figure B, we can see that the hard negative sampling strategy outperforms easy and semi-hard strategy, which further confirms our reasoning that using only hard negative samples can facilitate the model to learn directly about functional relatedness. Here in figure C, we can see that the use of both positional and distance embedding yields the optimal performances. The above results have demonstrated that it is not just because we have used deep learning methods, but also our model designs are critical to GRANA's effectiveness. Here, we have applied GRANA to an application of predicting the replaceability for a humanized yeast network. There are in total of 411 human yeast orthologous gene pairs, of which 174 are replaceable. This task is also a binary classification where positive means replaceable and negative means not replaceable. We have evaluated GRANA pre-trained, which refers to the model pre-trained on our own dataset, and GRANA fine-tuned, which refers to the fine-tuned model on this new dataset. We have also included eTNA. It is not surprising to see that eTNA as an unsupervised method achieves a near random performance. And even not fine-tuned, GRANA achieves a better than random performances, which further shows the advantages of supervised NA methods. And when fine-tuned on this new data set using cross-validation, the GRANA's performances can be further boosted. Here is here's an example of the experimentally determined replaceability of proteasome complex, and here is the GRANA's prediction. Again, this is another example of the experimentally determined replaceability of CCT complex, and this is GRANA's prediction. So overall, these results demonstrated the applicability of GRANA for extending the network alignments to empower other functional analysis of genes and proteins. In conclusion, our work is motivated by Tara and Tara++, the series of supervised NN methods. We propose GRANA, which is also a supervised biological network alignment, which is also a heterogeneous GNN-based framework. And through our experiments, we have shown that GRANA outperforms state-of-the-art NN methods in experiments. For future directions, we're looking forward for GRANA to integrate more diverse set of entity and association data, and also we can extend GRANA to other NA problems such as multi-species network alignment or temporary network alignment. In the end, I would like to thank my co-authors, Dr. Yunan Luo and Dr. Sheng Wang, for the help, and I would like to thank our funding agencies for the support throughout the research. This summarizes my presentation, and I would be more than happy to answer any questions if there are any, or if the time allows. Thank you so much.
Thank you. Thanks for a nice talk. We are slightly running out of time. Is there one quick question to take? Yes. Okay. One question. Yes.
Okay.
Niall?
Thanks for the talk. I'm just wondering, because what you presented by selecting the top scoring predicted pairs of proteins across the PPIs doesn't seem to be a real alignment, because alignments tend to be constrained to be 1-to-1, or you try to minimize the number of proteins to which you map one protein to the other networks. Also, often you try to maximize the number of proteins from the small network that you map to the larger network. So if you haven't taken that into consideration, I mean, I'm not sure the comparison with the other aligner is fair. So I don't know if you can comment on how you selected your protein pairs, maybe.
Right. Thank you so much. This is a great question. So let me see if I can answer this in two aspects. So first of all, I believe the one-to-many mapping of network alignment and the many-to-many network alignment are two separate subfields of network alignment. And it really depends on how you define network alignments. Because here in our work, we define network alignments as whether or not they share the same geoterm. And oftentimes, if we use this criteria, there will be many-to-many mapping instead of one-to-one mapping. And therefore, my second point is for all our baseline methods, we have used methods that perform the many-to-many mapping instead of the one-to-one mapping network alignment methods. For example, there are methods called JADEVO or other kind of one-to-one mapping performance methods. So I'm not sure if this answers your question, but so for each many-to-many network alignment methods, we selected the top predicted pairs and evaluated the Jakarta index.